Hello, Instagram. Today I'm very excited to have Merlin Sheldrake as my guest. He is a researcher, an author of this incredible book, Entangled Life, and an all-around mycophile, musician. He's a man of many talents. I'm super excited to get to really just nerd out with him and dive super deep into the world of mycelium and where the science of mycology is going, a little bit of the philosophy. I think we're going to talk some about sort of our shared backgrounds and interest in yeast and fermentation, and maybe get into a couple of projects, whether they were successes or failures. I want to hear, all sorts of stuff about what Merlin's done. And in his book, he describes sort of clandestine cider making from the apples that fell from the tree that supposedly inspired Newton to come up with the theory of gravity. So I bet he's got a couple of good, interesting fermentation stories, and I'm hoping we'll get into that. So the way that this is going to work and sort of the format of how I do my lives is I will keep an eye on the chat, but generally we're not going to do question and answer stuff. It's going to be Merlin and I talking to each other. And then after about an hour, I'm going to shut the live broadcast down. I'm going to try to post the video, and then Merlin and I will come back for maybe about 20-30 minutes of Q&A with the audience. So let me see if I can get Merlin on the horn here. And there he is. Perfect. It's exciting. I'm really excited. It's not every day you get to meet your mushroom heroes. And so I feel very, very lucky and very blessed that I have a chance to speak with Merlin here. So, hello.
Hi.
Hi, Merlin. How you doing?
I'm doing good. How are you?
Doing all right, sort of waking up slowly a little bit, but coming into consciousness and very much sort of just telling people how excited I was to talk to you, because I don't think it's every day that you get to actually meet your mushroom heroes and have a full conversation with them. So I came about you because my friend was like, hey, have you ever heard of this guy Merlin Sheldrake? He's really into mushrooms. And I was like, I don't know, man. There's a lot of people into mushrooms these days. I don't really know what you're talking about. He's like, no, he wrote a book. He's very legit. And I was like, oh, okay. So I started Googling you, and I saw you were a researcher. I saw you were an academic, and I was like, oh my gosh, I need to read his book. And so I went up and got it and thoroughly enjoyed Entangled Life. It got me thinking a lot. And the other thing I was doing was sort of interesting is I read a lot of this book live on TikTok, which I don't know if you're familiar with that platform, but it's an interesting way to reach a new demographic is what is probably the base thing I can say about it. And it really spurred a lot of interesting conversations and discussions and thought for me. And I know I sent you a very long email with a lot of stuff in there that we're probably not going to get to all of it, but I wanted to say thank you so much for being here. And I guess very quickly, did you have sort of an inspiration or something that led you to wanting to write this book? Was there like a crystallizing moment where you're like, I know I have to write this?
It kind of approached me bit by bit. I feel like the book found me. Someone said, do you want to write a book? And then I said, well, no, not really. And then someone else said, maybe this. And it just sort of, it built up bit by bit. And in the end, then I was like, okay, now I've got to do it. And it's definitely found me. I've tried to dodge it for a while, but it's kept coming back. And I have to look it in the face and take it on. And so once it got to that point, then I was we're just doing it. And it was driven by my interest and curiosity in the fungal world. And also the feeling that these stories about these organisms and these discussions about these organisms need to reach a wider audience. And so it really was trying to lift the lid on some of these topics and make them more accessible.
Was there, as someone who's having to write academic papers and know what a slog it can be to get through publishing data, after I finished my PhD in 2016, and I just had a paper published this year that was supposed to be like the final chapter of my thesis, and so it took 4 years. So familiar with a slog, but did you feel like there was some synergy there between what you'd been writing for your dissertation or for your publications and what you ended up putting in this book?
Well, in the sense that a lot of the material from the book came from my time spent doing research on fungi for my in my more formal academic work. But the writing couldn't be more different. When you're writing academically,
yes,
as you know, it can feel like, it often feels to me like I'm a dog howling at the moon. You're just throwing this stuff into the kind of a notch on some kind of academic post to prove that I've accomplished this, but I don't really expect anyone's going to read it. The idea that anyone would fight through this turgid prose that struck me as unbelievable. So it always felt a little bit defeating to be working so hard on academic papers. And so it was a great relief to write this and to really be thinking about the reader and being a good host, making sure that inviting readers into this space and really attending to where their interest might be going, or where they might be wanting to go within the subject. So it was a totally different experience.
I think I really like that feeling, because I definitely got that in reading your book, that I felt invited in. If someone had sort of, I was walking along in the countryside and I saw a cottage with a light on, and I went and knocked on the door, and someone said, "Hey, come on in, have a cup of tea. Let me tell you about mushrooms." So we don't have a ton of time. There's a lot of things that I want to dive into with you. So there's a quote that really stuck out to me, and I want to read this because hopefully it'll be the start of sort of an ongoing discussion between us. And that was, "Mycelium is a way of life that challenges our animal imaginations." And that was something I really liked, because you talk about mycelium sort of unlike anyone else I've heard visualize it before. Paul Stamets has said the famous quote of mycelium is the internet of nature, which you in your book even say that is a gateway term and really fails to understand the complexity or the depth. And that was something that resonated with me, because I remember reading Mycelium Running and seeing that statement in like the front of the book and being like, I don't know if I can take anything else seriously in this book, because that statement is so reductive. And it's a good, like you said, it's a good gateway statement, but man, it just fails to explain the complete, utter difference that fungi have. Like they defy animal concepts. You talk about it in the book of, we are singular, we are finite, we are definite, we have ends of fingers, we have toes, we can count all of these things about ourselves, and we can walk around in the world. Fungi, on the other hand, have no capacity to walk. When we put ourselves in their place, it's really difficult to imagine that, because how can you imagine yourself being this indeterminate organism that lives in 3D through its substrate and is constantly growing, and yet somehow all communicating with itself? And I guess I wanted to get into a little bit of the idea of sort of fungal plurality, as well as the idea of we're very animal focused, and how do we put ourselves into like a fungal perspective. And so I guess I wanted to ask you, is writing this book, it's pretty clear that you were able to put yourself in the mindset of a fungus to some degree. Can you talk a little bit about your process for doing that, or things you learned by trying to empathize with fungi?
Yeah, much of the writing of the book was a process of trying to find ways into the subject, to find ways that I could allow my mind to go to these places. And if you just think about it from the outset, it seems impossible. But what I came to realize is that our minds are the most indeterminate parts of ourselves. We might have very determinate body plans. We have, if you're born with 2 arms, you'll end up with 2 arms short of an intervention. And so our bodies are very determinate and centralized, but our minds are less so. So when we have thought that if you could draw a picture of someone's mind, I often thought that like a portrait of a mind would look like something like a mycelial network, and mind maps look like mycelial networks. But the way that thoughts branch, you have several thoughts at once, that there's this sort of unlimited shape-shifting capacity of our thoughts, and the way that they can weave together or separate or meld or fuse or branch. And so I really felt like these, this is why I talk about polyphonic music as a way in for me, this way of exploring both many and singular. So when you have polyphonic music, you have this intertwining of all these different voices. Each of those voices is a tune in itself, but together they make a tune that doesn't exist in any one of those tunes. And so for me, the polyphonic music, listening to polyphonic music, allowing my mind to flip between the singular and the singular whole and the plural many that made it up, that was a really helpful exercise, and just kept me shuttling between those conditions. And it allowed me just to get more of a feeling for this mycelial condition, where you have a multitude of hyphal tips and a single interconnected entity. And so it's just a little game that it's not, who knows whether or not my mind experience is anything like what it's like to be a fungus, but it helped me deal with these concepts of singular and plural at the same time.
Yeah, I like that. I mean, I think within fungi, you talk a little bit about ideas of how signals and information crosses those networks, and it's not entirely clear. I think we've got some molecular evidence that clearly things are shuttling around within mycelium. You know, there's movement of nutrients and resources, and they can remodel their entire network based on having found a new food source, or, you know, troping away or from, you know, certain types of stimuli. I think there's something in there where you sort of said, you know, it seemed like they damaged one part of mycelium and there was another one on a plate right beside it, and there was no transfer, at least of like a volatile chemical, which is a pretty common mechanism in plants. To, you know, if a plant gets injured, it releases a volatile chemical and other plants around it fold in their leaves or something like that. And it seemed like with fungus, movement of things across that network might be almost more like a nerve potential, because it was moving much faster than just simple chemical diffusion through the mycelium. So maybe there was like a bouncing thing. And that kind of gets back at like there being some intersection between animal and fungi. And people are always asking, well, animals and fungi are more similar than plants, right? I'm like, well, that's true, but they're still very different from each other. And I guess I was sort of curious if you had any thoughts on like that idea of the relay of signals in mycelium almost being like a nerve potential, or if there's any evidence to that you've come across.
Yeah, there is evidence. So a researcher called Stefan Olsson did some work in the 90s in Sweden, in Lund, and he was studying these forages, basidiomycete networks that forage over large areas and form long-lived networks. And so he went next door to see his friend, who is a neurobiologist, and he was investigating in moth brains and pheromone responses in moth brains. And so he was using microelectrode rigs. And he asked this person if he could borrow his microelectrode rig and stick it in the mycelium rather than the moth brain and see what happened. So it was a straightforward, you know, translation of a neurobiological technique into a fungal media and fungal space. And
you get a lot of academics who are just like, I see that method, I want it, let me try it.
Totally. I think it's brilliant. And but it makes this brain fungal network comparison unavoidable, because this was literally a brain technique being transferred. And so he found that fungi did, at least these long-lived exploratory networks, they did produce these spontaneous action potentials that were responsive to stimuli. So if you put a block of wood on this fungus, a wood rotting fungus, then it responded with an increase in firing rate of these electrical impulses. And so he tested a range of fungi and found this was not uncommon. It didn't seem to occur in fungi that form these ephemeral puffs and don't really range very far, not very exploratory or long-lived, like molds. But so it seems to be present in those fungi with a need for it, which are those ones that explore over larger areas. And but very little work has been done since then. There's a guy called Andrew Adamatsky who's picked it up very recently, but he's been looking at the electrical potentials in entire mushrooms. He hasn't been sticking these electrodes into individual mycelial strands. So there's a big question mark over this, and I think it's one of the most exciting potential areas in fungal research. There's a whole new wave of research into electrical signaling in plants and bacteria, and since 2015 it's been known that bacterial biofilms, they use action potential like waves that pass across their whole colonies to signal nutrient status in different parts of the colony. So the main thing is that, to put it in the context of brains, is that our brains didn't evolve their chicks from scratch, and these properties of our brains reflect much more ancient properties of cells that were present many millions of years before formal brains arose, given that we find electrical signaling in plants, bacteria, fungi, as well as animals. So I think there's a lot of potential here to find out new things about how these networks coordinate themselves, potentially over large distances. And it could really transform our understanding of ecosystems.
That's cool. For me, from a scientific perspective, there's and I know you know this, but there's a concept called quorum sensing. And this is something that, you know, yeast and a lot of bacteria use, because it's sort of the idea of how many of my neighbors are around me. And so a lot of organisms, planktons, and a lot of things that grow in big swarms are often very exquisitely sensitive to the metabolic byproducts of their, you know, whatever else is in their environment, including themselves. And they're able to titrate their growth and change, you know, physiological states or metabolic states based on certain quorum sensing molecules. And for me, thinking about mycelium as this giant mass of life all kind of mishmashed together in the soil, you know, my first thought was, oh man, there's got to be some interesting quorum signaling going on, you know, within species, maybe within mycelium, and then between species. And I mean, the other thing too that sort of blew my mind, and you know, clearly I knew this, but it hadn't necessarily translated into active thought, was there's bacteria inside that fungus. So there's, you're taking the microbiome of the soil, but there's already a microbiome of the mycelium. As you know, then you have like endo versus exobacteria, and I'm sure there's things that shuttle and move between them. And you know, my dad has worked on rhizobium for a lot of his career and looked very intently at the association and formation of nitrogen, the little nitrogen fixing nodules, and how like the factors that go back and forth between the bacteria and the invasion and the transition from one physiological state to another kind of thing. So I've thought a lot about this in a nitrogen fixing context, but like that's, you know, legumes are 10% of plants on the planet, and fungi and mycorrhizae are sort of associated with the other 90%. And it was a fairly recent realization to me that I was like, oh my gosh, fungi are the glue that hold every form of life together. And your book kind of helped cement that for me, because I was like, wow, I hadn't totally clued into the fact that like plants coming to land was a function of fungi coming to land and enabling plants to move there. And that was just such a mind-blowing concept. So I guess I'm just kind of curious about if you have thoughts on like lichens being really primitive forms of early plants, and then like, you know, just that journey a little bit of how like fungi and plants kind of terraformed our planet a little bit. Yeah, I'm just curious to hear what your sort of impressions and all that.
Yeah, I agree. It's one of the most amazing stories in the fungal, in the world of amazing fungal stories. It never ceases to amaze me when I hold a plant to think that this entire lineage of land plants has arisen, and is the outcome of these mycorrhizal associations, and also the endophytic associations. All plants have fungi in their leaves and in their shoots, and so these really are fungi that have evolved to farm algae, and algae that have evolved to farm fungi, and both at once. You know, these arrows point in both directions, firmly at once. But there's a lot of discussion about the details, you know, because it's quite hard to resolve these earliest stages of this partnership. And so, for example, it's not clear whether the algal ancestors of land plants washed up with their fungal partners in tow. Lots of algae do have partnerships with fungi. Lots of seaweeds that you see on beaches are able to withstand that periodic desiccation because of their fungal partnerships.
Called like fungal endophytes within the seaweed, or
Psychomicrobiosis. It's a big deal.
I've never heard of, I have to go Google that.
Yeah.
Would you call it
Psychomicrobiosis? Yeah, it's a pretty under-researched area of this subject, and so it's very possible that the algal ancestors of plants washed up with symbiotic partners in tow. It's also very possible that they met the fungi on the shores already, and these fungi were living in this kind of soggy detritus on these damp shores of lakes and rivers. So it's not clear which happened first. With lichens, certainly with formal lichens as we know them today, it's thought that this is a parallel evolutionary story, that plants are not continuous with the evolution of lichens, that it happens separately and after the formation of land plants. It's very possible that lichen-like organisms existed long before, because it's quite easy for plants, for fungi and algae and bacteria to come together to form lichens. And it's happened many times, well, several times over the course of evolutionary history. So whether or not there were lichen-like organisms on land, it's very possible that they were. Kind of crust-like things before land plants is not known, but is a high possibility. But certainly the algae and fungi have a tendency to associate. And in the book, I described these great experiments done in 2014 at Harvard, where they get fungi and algae, the free-living fungi, and provided that both are in a condition like nutrient condition that one of them can do something that the other can't, they form young symbiotic associations in the course of 10 days or so. You can witness the birth of a new symbiotic association. So it's like two marvels that are released from the edge of a bowl, like these types of organisms want to associate and have done so repeatedly.
Circle around to meet up together. Yeah. That's an amazing concept. And I mean, that's another great sort of idea too, that living organisms have this capacity to adapt and to morph. And you see with fungi all the time that there's like multiple different forms, a sexual form versus an asexual form, and they look completely differently and have completely different life cycles and inhabit different parts of the environment. And there's something in there that is really interesting about what is symbiosis, what is mutualism, what is altruism, and from like a biological perspective, from a very traditional kind of taxonomic thing, humans really love to put stuff in categories. And we like to say, well, this thing is parasitic, this thing is symbiotic, whatever it is. And fungi in general love to defy those categories because fungi are, like we've talked about, they are undefinable. They are not discrete. And so they work that way in life too. They're not going to just do one mode of metabolism. You know, morels can switch from being mycorrhizal to being saprophytic. And there's, you know, so many fungi out there that can do asexual as well as sexual, sexual reproduction, and just sort of mind blowing. And for me, one of the most interesting things was when I was interested in mushrooms as a kid, kind of forgot about them for about 20 years of my life, where, you know, I went to college, I got into home brewing. And that got me into yeast, which took me to my PhD and a whole bunch of wine and fermentation stuff. But it wasn't until after I finished my PhD and I finally had a little bit of mental brain space to think about something else that I was like, oh, I'm going to go for a hike and find some mushrooms. And finding mushrooms was really cool. But man, finding Mycohetereotrophs, seeing those plants that, if no one's not familiar with Mycohetereotrophs, these are non-photosynthetic plants that grow in association with fungi and a host tree. And the sort of quick and dirty of it is that Mycohetereotrophs have always been viewed as parasites, that they are leaching carbon from the mycelium. And that's really only been what's attributed to them, right? They're parasitic. But recent research has shown that there's a far more complex, far more dynamic relationship between the flow of nutrients, between these Mycohetereotrophs, between the fungi, between the host tree, and how that changes as a function of seasonality and other environmental factors. And that was so cool to me. And Merlin literally did his PhD on Mycohetereotrophs. So would you give me kind of the elevator pitch version of what you worked on in grad school?
Yeah, so I was working on these long-term fertilization plots and nutrient gradients in tropical forests in Panama. And the nutrient status of a soil has long been hypothesized and has been demonstrated to have an effect on the trading relationships between plants and their mycorrhizal fungal partners, because the theory is that the plants are making considerable investment, carbon investment in their fungal partners. And if you add lots of phosphorus, then the plant doesn't need those mycorrhizal fungi harvesting phosphorus and transporting it back to the plant. And so the plant can reduce its investment in the fungi, and it changes the mycorrhizal situation. And so I was looking at how these different nutrient regimes were changing the balance of the relationship between plants and their mycorrhizal fungal partners. And I became interested in Mycoheterotrophs because these fully mycoheterotrophic plants, there's no way for them to produce their own carbon because they have no chlorophyll there and no leaves. And so they are getting their carbon via a mycorrhizal fungus from another plant. And so wherever those exist, there is a reliable flow of nutrients and carbon and water from one plant to another through a fungal network. And I wanted to see whether or not changing the nutrient regime would affect the Mycoheterotrophs. And I found that they were very sensitive to phosphorus and across a big gradient. And there was a cutoff point of about 2 parts per million where Mycoheterotrophs could or could not exist. And you find them there, and then you find them not there on the other side of this threshold, which suggested some but the fungi they depended on were still there at high phosphorus. So it suggested that the phosphorus was changing the trading relationships between mycorrhizal fungi and their plants in such a way that the fungi were no longer supporting these Mycoheterotrophs. Not quite sure why, but it opened this whole kind of inquiry. Anyway, I became fascinated by these plants because I spent days counting them, looking for them, and the more you look at
your field work was like you literally on dirt. You know, I've talked to people who had some tough field work, but that sounds, you were like inches away from the dirt. So it's not surprising in some ways that you were able to then think about things from that fungal perspective, right?
Yeah.
So can you tell me a little bit about that organism that you were studying with? Voyria?
Voyria.
Oh, okay.
It's in the gentian family. White stalk, Voyria tenella was the species. White stalk with a blue flower. With just these little scales where its leaves used to be. And this funny, clumpy, star-shaped root system, very reduced roots. And when you look at them, they're really just these giant cavernous spaces where they basically farm the fungi. And they have a particular style of growth where the fungus grows in. One of the Mycoheterotrophs dilemma, as described by Stefan Imhoff, a great Mycoheterotroph expert, is that they need to have reduced roots to shorten the distance that carbon has to travel before it gets into the plant, but they have to be big enough so they can attract fungi. And if they are too small, they're not going to be able to attract enough fungal infection to support them. And so Voyria tenella has these cavernous spaces where the fungus grows in, and then it has these sort of in the central space in the roots, then the fungus grows up and down and kind of lives, sort of exists there in its mycelial form, and it grows down from the inside back into the tissue of the root, where its cells, its hyphae, swell into these kind of balloons and then pop and are digested by the plant. But by having this interior space where the fungus can live in these mycelial form, the plant is kind of hosting the fungus rather, and making the most out of the fact that there might be only 1 or 2 infection points. So it's got this very highly evolved fungal farming method, more so than some other Mycohetereotrophic species.
That's I had the mechanism of how the nutrient exchange happened. I had no idea about it. It reminds me a little bit of what I was talking about with those nodes where the bacteria are invading and they're sort of setting up a very particular environment for an organism to do this, reprogram its metabolism or forcing it into this type of relationship that might not seem necessarily symbiotic. It might seem like you're sort of giving more than you're taking potentially. And I guess with that in mind, I'm curious to hear, biologically, I was saying that Mycohetereotrophs had been defined as parasites. And in reading some, particularly about the relationship between Sarcodes sanguinea, the snow plant, and this Rhizopogon species here in California, I'm pretty convinced that it sounds like there is mutual benefit on all sides, and that it's not just that the mushroom is, symbiotic with the tree and the Mycoheterotroph is just parasitic. There's flow of nutrients between all of them, and there's benefit that they can all bring to each other. So I guess I'm curious if you saw that in your system, and then what other sort of thoughts you have on parasitism versus sort of altruism with.
I'd be interested to hear what this research about the Rhizopogon and this.
I'll send you a paper.
Yeah, that'd be good. The parasitism hypothesis has always been this. The way they've been understood, although people admit that it's very difficult to test whether this plant is having a detrimental effect on the fungus. It's very hard to assess fungal fitness experimentally, mainly because it's very hard to actually grow these plants in a greenhouse. Yes. And I had some very, very limited success, but it's incredibly difficult. And you're just trying things out. You're just throwing stuff at the wall, seeing what sticks. And so. But there are other possibilities. So there are different types of Mycoheterotrophs. So in some ferns, like Ophioglossum, which has different, has alternating generations. So it produces what's called a gametophyte, which is this little puddle of tissue, which lives out of the light. It lives buried in, covered totally by whatever's growing around there. And so it doesn't photosynthesize at all. And it's supported by a fungal connection with the sporophyte generation, which produces the spores. And so it's a kind of parental care. The sporophyte supports the next generation in this phase of its life cycle. And then the gametophyte gives rise to the next sporophyte, which can photosynthesize and support the subsequent gametophyte. So their way of doing mycoheterotrophy, these ferns, is very much it's a kind of a strict parental care. It's as strict parental care as I've heard of in the plant world, and so this is clearly not a parasitism in the conventional sense. And there are other ways too. I mean, there are lots. All orchids, for example, start off their lives as mycoheterotrophic, as pulling carbon from fungi, whether they be saprophobic saprotrophic fungi, and or mycorrhizal fungi, and they can be both. And so. But then a lot of them, when they get older and they're able to photosynthesize, they have more highly developed leaves and photosynthetic apparatus, then they start to return the favor to the fungus. This is the what some people call the take now, pay later hypothesis. And so many plants do this, and it's possible that mycoheterotrophy evolves from these what's called initial mycoheterotrophs that did this so that it could get a foothold in these heavily shaded forest understories. Where it's really hard to get going if you're a young plant in particular. And so in these cases, if the plant fails to repay, then it's defaulting on the buy now and the take now, pay later thing. So it's rather than being a sort of full-on parasite, it just hasn't paid its dues. So you can see these different ways that you can approach how mycoheterotrophy could have evolved in a way which is sort of graded, and it's not just straightforward. screwing over of the fungus. But I think a lot more work needs to be done. There are many other ways that the plant could be supporting the fungus without being part of this nutrient economy, without repaying in these conventional currencies that we look for. Possibly by shelter, for example, being given sanctuary, a kind of refugium in the wilds of the soil and in these cavernous root spaces. That's one possibility. But I'd be keen to hear what was the Sarcodes giving to the rhizopogon.
I mean, again, I haven't read too much about this, but I think the idea was that where the snow plant was present, the rhizopogon had a much wider network. So it was one of those things that when the microheterotroph was there, the fungus had a wider reach, and it would sort of, it was creating mats of, fully myceliated forest and soils, which, is always going to be better to have sort of more mycelium because there's a lot more nutrient holding capacity. The soil is more, locked in place. And the interesting thing for me was that the snow plants grow from rhizomes. And so, I think of a rhizome as a little bit like an underground battery. And so this is just my personal sort of thought and theory, was that potentially, California deals with fire ecology, and a lot of our state is very much evolved to have fires move through. Right now, it's a little out of control because there hasn't been burns for a long time, and there's a lot of invasive species that are creating too much plant matter, so the burns are happening too often, too fast, too hot kind of thing. But in general, a lot of the plants here in California are evolved to some level of fire moving through the environment with some periodicity. And when you have a fire move through, you lose a lot of the initial fungal diversity, at least in the top layer of soil. And it returns over the matter of a couple of years, but initially you see a drop in biodiversity and then it kind of comes back up. But one of the things I was thinking about was that maybe these rhizopogon Alinea, whatever it is, are associated with the rhizomes of the snow plant. It's acting almost like a battery or something to like recharge the fungus. If the tree has dropped a bunch of its needles and is not doing so well, maybe these little snow plant batteries can recharge the fungus or help them get through, or at least repopulate those top levels of soil. And that is me just postulating stuff. I can send you the actual data and you can see whether or not that seems like a reasonable hypothesis. But it was just one of these things that really blew my mind, to be like, it's not just the flow of nutrients, but maybe it's also the way that you approach the environment. And that these things, if you're talking about a network, these mycoheterotrophs are acting like nodes in that network, where you can then explore, you can go have refuge and then explore new environments or colonize new areas. Which again, yeah. I think, sorry, one thing, just you talked about the idea of like, it's really hard to grow stuff in a greenhouse, but that's because so much of science has been done in isolation, and yet everything happens in these communities, and we're not even like, sorry, go ahead.
Totally, no, it's exactly right. And you have to somehow assemble in a pot the entire community. It's a bit like the difficulty that people have with getting truffles to fruit. You can inoculate tuber magnatum onto the roots of a tree, but getting it to fruit requires a knowledge of its sexual life and its environmental needs that we simply don't have. But I like your idea that the Sarcodes as these kind of staging posts for this fungal network. I'm not sure. I just haven't heard of anyone studying the degree to which fungi can grow out of these roots. When I was looking at these Voyria roots, and Stefan Imhoff, who I was working with, he spent a lot of time looking at them under a microscope. I always talk about them as these fungal hyphae as where they are leaving the root. He's like, no, that's where they come in. They do not come out. When they come in, you can see exactly what happens. They get trapped within this root space. There they live, and they pop and they release their contents, but they're not emerging from this space. It's a one-way street. So that's how he thought of Voyria roots. But I'm not sure about Sarcodes. They all have very different habits. Sarcodes, for example, partners with not with a vascular mycorrhizal fungus, with a Basidiomycete, and so it could be a different style of relation. Maybe the fungal partners are able to grow out of the rhizopogon, out of the Sarcodes root. But who knows? I think it's a fascinating question, and to broaden our scope to try and see these different kinds of benefit that might be provided. We get very locked in, certainly in the mycorrhizal sciences, we get locked into these nutrient exchanges, and it's easy to become small. Narrow your focus onto this and see it very transactionally, but I think there could well be some other effects.
Yeah, I mean, just looking at ecosystems as a whole, and I sort of brought up the idea that like I'm realizing that fungi is, I know this sounds sort of like a weird hippie hand wavy concept, but like fungi is the glue that literally holds life together. I mean, we have a food web, we have a carbon and nitrogen cycle, and if we didn't have fungi, we wouldn't have those things. They are
Mm-hmm.
they are the keystone of how organic matter is broken down, and also how so much of life is able to function in potentially detrimental conditions, from endophytes and arbuscular mycorrhizae and all sorts of different things that are subtly helping plants and animals sometimes adjust to environments where they would otherwise not have a foothold, kind of what we were talking about with that first colonization steps of land potentially. And two, getting at the idea of nothing really being in isolation. My PhD, was basically looking at a phenomenon in yeast where there was a physiological shift, induction of something called a prion state, but it was basically kind of an epigenetic switch that changed their capacity to prioritize carbon sources. So normally yeast are super focused on glucose, and this was an epigenetic switch that caused them to be less focused on glucose, which made them less dominant in fermentation, which from like a winemaking perspective wasn't good, but from the yeast perspective was potentially a great thing if there was a lot of competition around. And this is what we saw a lot of, like lactic acid bacteria, acetic acid bacteria, that kind of thing, could shift fungi into a different physiological state, shift the yeast by inducing this prion state. And suddenly, instead of being obsessed with glucose, they're like, oh, cool, we can eat any carbon source and we'll be more of a community member rather than this sort of dominant thing. And so, again, the more I learned about mushrooms, it makes sense too that fungi have different modes of whether you're trying to be dominant or I'm going to sit back and be a community member and just sort of exist. And a lot of my project was realizing, I was like, oh my gosh, almost all of molecular biology has been done completely in isolation. And the really fascinating thing is when I started looking at the frequency, the inherent frequency of this ability to induce this prion state, it was very different among different niches of yeast. So wine yeast had a very high chance of going into this, what it was called the GAR plus, the glucose-associated repression, but prion state, whereas like a beer yeast or a clinical lab yeast had a very low percentage of going into the state. And that's because they were exposed to very different types of environments. And clearly, if you're in a mixed microbial environment, there may be a benefit to maintaining this kind of cryptic prion, shifting ability. And so that really got me thinking about communities and whether they be, in a little fermentation or they be out in the soil. But you are, as far as I can tell, a prolific and enthusiastic home brewer and fermenter and tinkerer. Can you tell me a little bit about if you got any like fermentation projects going on or like things that have inspired you with beer or wine or all of the above?
Yeah, it's a fascinating world, and it's just been endless. Like so much of fungal inquiry, it's just this the corridor after corridor with door after door, and your interest is the ever deepening curiosity, the sense of just sliding deeper, and so many people have this experience in the fungal world, and it's really thrilling to meet other people who are similarly engrossed. And so fermentation is one of those. So for me, it was many things, but it was, as I came to do it more, I realized that it was a way of noticing these big metabolic transformations that take place in the planet that really structure the planet, these chemical weather systems, these biogeochemical cycles within which we live, without which we couldn't live. And it's so hard they're kind of abstract to us. You can draw a schematic on a piece of paper and you can look outside and know that it's going on, but it's hard to really, really get it. And so I realized that fermentation was a kind of domesticated version of a biogeochemical process. It was a metabolic transformation involving a whole community of organisms taking place in succession of stages that I could actually taste with my mouth. I could taste the chemical difference between each stage. And at the end, if it's an alcoholic drink, then it will change my perception of the world. So it's like a way of becoming really to really be able to notice these metabolic transformations in a desktop form, and through that become more aware of what's going on in the soil, what's going on in the atmosphere, what's going on in plant animal microbial bodies. And so that's a sort of one of the ways that it's been fascinating to me. At the moment, I've been so busy talking about fungi that I've had so little time to actually do fungal things, but I do have I've been working on these pickled brined lemons. Make an amazing, just very simple, very stable ferment because they're quite high salt, but they produce this amazing thick. What was the juice becomes this thick yellow amazing flavored sort of relish that you can add on anything. I've got some sloes. I don't know if you've sloes in the West Coast. They're kind of wild plum, but I'm trying to do those in a umeboshi.
I've heard of like
ferment as well.
slow gin kind of thing.
Exactly, yeah. So I'm
okay.
trying an umeboshi slow recipe.
Oh, cool.
Well, I haven't had a recipe. I'm just adapting an umeboshi recipe.
Yeah, I've had a lot of plums before. I'm like, should I umabashi those? I think someday I'll get around to it. Droops in general are good.
Yeah. So that's going on. Hot sauce is a perennial favorite. I do a lot with hot sauce. My brother fermented hot sauces. There again, they're really great because they like long ferment, quite high salt, and they are stable at room temperature when you ferment them, so you don't need to store them in the fridge. But these are just these sort of things that are always going on that are very easy and basic necessities, the basic provisions. And then there are more exploratory ones, of course, and there have been many of those.
Have you? So you mentioned in your book, and I was really excited because I think back in like 2007, I took a trip. I did like a summer abroad trip to Spain, and we had a project to write on some aspect of food production. And I was really into beers. I'm going to write about beer, and I stumbled upon the idea of gruit beers, which really blew my mind. And if people don't know, there's this German brewing purity law called the Reinheitsgebot that I can never pronounce quite correctly. But it was essentially a political ploy in the era of Calvin in Germany that was separating the German church from the Catholic Church. And this German purity law came about because they're like, well, beer is only going to be brewed with water, hops, barley, and then they didn't know what yeast was, but that sludge from the bottom of the bucket kind of thing. And it put an end to the age of the gruit beer. And gruit beers is traditionally, through much of history, is what people were brewing as beer. And this was people trying to make beers and get some of the preservative properties that hops lends to a beer, mostly as an antioxidant and as an anti-spoilage type of thing, because beer was probably the predominant beverage for a very long time, low alcohol beers. It was one of the only safe liquids we could drink. So, I mean, through a lot of human history, we were probably at like sort of a steady buzz, 1, 2, 3% kind of thing on like a daily basis, which interesting when you look at history through that lens. But you mentioned actually doing some gruit beers and brewing some things where you were using, I don't know, swamp rosemary, or what kind of botanicals did you play around with when you were making those beers?
So I was using, for the gruits, I used bog myrtle, Myrica Gale, and wild rosemary, which is, Labrador tea, which you have in the States, and yarrow. And it produced these, they add plenty of bitterness, plenty of complexity, plenty of preservative. But it was just a different kind of experience. Hops are, you can use hops, as you know, to get to sleep. They're a way of relaxing, a kind of herbal sedative. And so if you drink hop beers all the time, I have a problem with these really strongly hopped American West Coast IPAs.
Yes,
it's like trumping blast of hop. It's just a bit over the top. I try to get away from the hops a little bit because, they're a cousin of cannabis, and you can kind of feel this sort of sedative effect. At least I've noticed that. And so the gruit beers, it was just a different thing. It's much more up. It was much more a lively intoxication, and people talked more loudly. It was a really fascinating flavor. I definitely was a fan of the gruit beers. I also made nettle beers a lot. And I thought that, people would make these beers with whatever plants were around. And moreover, what plants did you want to incorporate in your herbalism? What plants did you want the medicinal properties of? Well, you put them in a beer to preserve those medicinal properties. So if you wanted to have nettle medicine in a seasonal period where the nettles aren't there, then the nettle beer would allow you to have the nettle medicine then. So to think about it like that, and to have so the nettle beers are always a favorite. I love those. They're very savory and rich, and full, and I can never get enough of them. It feels almost like a food, very nourishing.
And that's through a lot of history. That's what beer was. So you mentioned something that was really interesting. You're talking about the secession of organisms, and that's, I think, a thing that a lot of people don't necessarily realize is many fermentations. There is a primary alcoholic fermentation with yeast. But then even with like red wines, there's a malolactic fermentation where Enococcus inii will convert the malic acid, which is a very, very sour fruit acid, sort of green apple-y, into a rounder lactic acid, which is gives you a little bit more mouthfeel. So one of the classic things that I learned about was sour beers. And at age 7 or something, I was in Brussels with my dad, and he let me try a little sip of Creek. And that moment changed my life because A, I was 7 and I was having a sip of a beer and I felt really cool. But it completely shifted the way that I thought about fermentation and drinks and anything, because I was like, oh my gosh, this is such an incredibly complex and insane process that my dad described to me, this multiple succession of different organisms. And one of the ones in sour beers that's really important is a yeast called Brettanomyces, which is named Brettanomyces because it was isolated in Britain. And it is one of the classic sort of beers. In beer, it gives, I love the flavor of Brettanomyces in beer. In wine, I hate it. It ends up tasting like smoke and horse blanket and a bunch of characteristics that I'm not a fan of. But bready beers are something I absolutely love. So I know you've done some stuff with wild yeast. Have you run into Brettanomyces at all, or is that a yeast that you're familiar with?
I'm not actually, but I do love these lambics, and I've just been drinking Gers all the time, these blended lambics, and it's really my favorite thing. And the complexity, the way that the flavor unfolds, it just feels like a world in there, and one that could be explored in so many ways. And because you're always you're collecting the yeast wild from the air, then wherever you make it, you're going to have a different kind of community and a different way of assembling. And so I'd be really interested to try doing some English lambics, and maybe the Brettanomyces would be one of the really popular attendees at their party, given its name. But I haven't heard of that one before, or read about it. But it makes sense that there'll be a huge number of different yeasts in these. But first of all, because it's falling out of the air, and although some might rise to dominance within the process of fermentation, still you have a lot there. And as these different phases of fermentation unfold, then they'll rise to prominence. And of course, when you're in the creeks, when you're adding this cherry juice, that's a whole secondary fermentation that will undergo with a whole different kind of community. So yeah, I love these guys. Really seem to it feels like a priority in the process to maximize the different types of yeast. It's like maximal diversity of the yeast, maximal connection with where you are by having these big shallow dishes and the roofs and the rafters of these breweries.
I mean, I've heard it described a little bit as like, it's someone playing a trumpet versus, a whole jazz band, or, the single violin versus the whole orchestra. And that's exactly what you're talking about. You're getting all of that extra complexity and stuff. So.
Exactly. Let me just plug in my phone.
Okay. Life happens sometimes. Okay, so yeah. No, sorry.
No, you go. Yeah.
So, yeah, I'm just being aware that we have like 13 minutes left, and there's still like a whole page of things I want to ask you, but I'm going to try to pare it down here. So one of the things that really, I don't think it was mentioned explicitly in your book, but my brain has just been dancing around the concept, is the idea of what I'm going to call a trophic transformation, especially by fungi. So if we think about like trophic levels in nature, we talk about stuff like, algae being on the very bottom, and then small fish, and then a bigger fish, and then, a tuna and maybe a shark kind of thing. And every time you go up in trophic levels, you're losing a lot of efficiency. And this is one of the reasons that something like beef is so environmentally expensive, because we go up a bunch of trophic levels and we're not getting a lot of return on our trophic investment. And something that I've seen that fungi do is they do, instead of jumping trophic levels, they're sort of going sideways. They do this trophic transformation of organic matter into things that make it more valuable. And what I'm talking about is stuff like, just like fermentation, right? We can take sugars from a plant and ferment them into alcohol, which becomes this substance that is inherently safe to drink. Usually when something's been fermented, there's no longer a food safety risk. And, alcohol has effects on us, which humans seem to like, for better or for worse. But, it's a transformation of that. There is stuff like miso and soy sauce, where fungi have changed and made stuff more bioavailable. I've seen incredible technological things that are happening now, where people are taking like the textured vegetable protein that goes in all those vegan sausages and meatless burgers and whatnot, and they're fermenting that with like shiitake mycelium to make the protein content, the amino acid profile, better fit what we need as animals and make the nutrients more bioavailable. And so this is, it's just amazing to look at sort of the trophic transformation of fungi. And I guess I'm just curious, as as another fellow mycophile, do you see that being something that you think maybe we should like explore more of, and see if we can find foods that can be trophically transformed by fungi?
Yeah, well, and it's also one of the ways I think that we've related with fungi for the longest, whether it's in alcohol, which is extremely ancient, or as any other yeast food like bread. And there are so many others. Miso, as you mentioned, is a huge one in East Asia, and these ways of preserving. To preserving, you don't want to digest this food. You don't want to go through this whole process of being respired, but you want to horizontally transform it, as you said, into something which is going to keep. And for most of our lives, most of our evolutionary history, they have not been fridges. And so we've had to recruit the services of these transforming organisms. Fungi are big ones, bacteria, of course, another big ones, but fungi are a big part of our evolution and our cultural evolution, our biological evolution. And so I don't think there's any option for us moving forward. I think this is just, they're here to stay, and this question of how ingeniously we want to innovate here. And I really do, I think these areas like adapting the corn meat substitute with the shiitake mycelium, I think these are fascinating areas of inquiry and really exciting to get these meat substitutes up onto a whole new culinary level, because the possibilities for helping to alleviate the crisis are huge, obviously, and potentially great health benefits as well. So I'm all for it, and I think that there could be a lot more experimentation done like this. And it feels like we really are at a dawn of a new era of experimentation with these technologies.
Yeah, they're definitely on the cusp of something big. And I, just recently I did a little video about wheat lecoche, the corn smut fungus, and realized I was like, holy cow, that's another example of this sort of, trophic transformation where we've been viewing this thing as a disease, as a nuisance, and really it's far more valuable than corn. I mean, we should be cultivating this. And I know there are some people here in the States, I think, who put money into trying to cultivate it. And there's some pushback because people are concerned about equitability and access and stuff like that. But I mean, it is amazing that I'm really looking forward to be able to buy a little vacuum packed thing of wheat lecoche in the store pretty soon and be able to cook some delicious stuff with that. And I think, I would love that kind of thing. One of my big things is if we could get the world to cut their meat consumption by half and eat mushrooms. And I'm not a vegetarian, I'm not a vegan, I love meat, but I recognize that one of the biggest things we can do to reduce our environmental impact is eat less meat. And health-wise, it makes a lot of sense to eat more mushrooms because they're really good for you and they're really tasty. So I kind of love this idea of trophic transformation too, even within our own lives, that, maybe we'll just cut out some of the meat and I'll add some more mushrooms. And I personally, since COVID started, I've dropped a bunch of weight, and it's partially just because I've been eating more mushrooms and kind of cooking from home sort of thing. And so, it's putting into practice for me, oh yeah, this is real. This can happen for people. Okay, but the last thing I really wanted to talk about that you brought up in your book that I thought was super fascinating was the idea of what I think Richard Dawkins proposed as the extended phenotype. And this came a little bit from the idea of a cordyceps infects a bug, and then somehow this bug, controlled by the fungus, is still able to move through the world and recognize stuff like light and gravity and, see things, even though a fungus has no eyes, no brain, no mouth, no sensory organisms, that we recognize from an animal perspective. And even the idea that maybe getting drunk off alcohol could be an extended perspective. And that kind of got me thinking, I was wait, if everything has a fungal endophyte and almost every plant has a fungal partner, arbuscular or otherwise, is the world not just an extended fungal phenotype kind of? And this is where my brain broke down. I was I gotta ask Merlin about this.
It's a good question, isn't it? So it's, as I talk about in the book, Dawkins kind of unleashes this concept, and then I'm a little bit concerned for what he's done. He has to start placing stringent limits because he sees it getting out of control. So in the glossary of his book, he describes the extended phenotype as all the effects of a gene on the world. And obviously this is a very loose sense. And so, yes, so it's the effect of the alcohol producing genes in a yeast. They produce alcohol, and then the effect of that alcohol is to get humans drunk. And so the intoxication of humans, in this sense, could be seen as part of the expression of the yeast's genome. And yeah, that's true. But I think if we talk about it from Dawkins's point of view, I think he would argue that this was too loose, this was too extended, an overextended phenotype, because for Dawkins, at least, if the extended trait has to have some bearing on the fitness of the yeast. So it doesn't strike me that our intoxicated state, the quality of our intoxication, directly impacts the survival of those yeasts that produced it.
But does it? Because I mean, I come from the wine industry where people are like, I have my favorite yeast strain, EC11. I always remember EC1118. Oh, when I do whites, I do that. I mean, maybe there's something there.
No, I see what you mean. But perhaps it's more to do with flavor than the intoxication.
Yeah.
Do you think?
Yeah.
I mean, I certainly have noticed different types of intoxication with different home brews, and that's been something that's been very interesting to me. It's like, why do I feel a different kind of intoxicated on this compared to this? But it strikes me that certainly in the wine industry, in these places where people are actually selecting for yeast, that it's definitely seems to be more of a flavor thing. Although I may just be ignorant of these breweries who are doing complex bioassays, testing people's behavior. But no, I see what you mean, and I think you can argue it each way. I just to stick up for Dawkins's strict case that, but I think that maybe it's more helpful to think about it in analogous terms to the way that we think about like the reward that a flower provides to a pollinator. So a nectar reward, or the flesh of a fruit, these kind of plant rewards that encourage a certain behavior in the animal that then propagates that organism. So if we have the yeast produces alcohol, and then that causes us to cultivate the yeast, it doesn't necessarily say that our intoxication, the state of our intoxication, the quality of intoxication doesn't need to co-vary with the genetic component, the parts of the yeast genome producing the alcohol. But nonetheless, this alcoholness intoxication is what binds us into this relationship with yeast, no question about it. Although you could also argue that it's also the ability to preserve liquids that does it as well, or that we can't really separate those things. But no, I think it's fascinating. And the impact of alcohol on human evolution, cultural and biological. There's this mutation in the alcohol dehydrogenase gene, which happened 10 million years ago, so long before we became humans. But that allowed us to explore these new dietary niches, like this overripe fruit that had fallen from trees. And these small amounts of alcohol, which would otherwise be poisonous to us, were suddenly could be used as a source of energy. So we have this imprinted into our genomes. Our coexistence with yeast, and so I think it's something to explore. And the extended phenotype, I'm totally up for overextended phenotypes as well. I think it's a fun exercise to let these the impacts of a gene on the world cascade outwards as far as you can take it. Just to see what happens to your mind. I feel that that's quite a healthy practice of getting us to grow our minds, to grow our awareness of these organisms and their relationships to their environment. So I think it's a good. Definitely a good exercise.
Yeah. Well, I mean, certainly, we talked about sort of at the very beginning about putting yourself into the mind of a fungus. And I spent 7 years trying to put myself into the mind of a single-celled fungi that lives in a big tank of juice, and what would I do, And it's not necessarily the easiest thing to empathize with, because it's so different from what we think about. But it did get me thinking a lot about, did we select yeast, or did they just choose us? And again, that gets back to the idea of fungal intelligence, which is very hard to define. And like you said, there in your book, you talk about some parallels between the idea of like networks and brains and intelligence, and it's there, but it breaks down very quickly if you start thinking mechanistically about, actual intelligence, our consciousness versus what a mushroom does. But if you shift your perspective and you be less animal focused and maybe more fungal focused, I think it's very possible to recognize that fungi have this sort of innate, inherent intelligence, and that maybe they've been guiding humans a lot longer than we think.
I mean, there's a good study that I read recently where it found that different types of yeast attracted on rotting fruit, attracted fruit flies to different degrees. So fruit flies had favorites, and the favorite yeasts were dispersed more readily by the fruit flies than the less favorite ones. And so it's very much like this plant pollinator interactions. And so if yeast has been attracting animals, whether human or not, for a long time, then we'd be very part of this big story where alcohol and the volatiles were the yeast's way of getting around. For sure. And with regard to the intelligence thing, I mean, I think there's the debate recently and intelligence has become much more broadly defined, thankfully. And rather than be defined in strict human terms as something that requires a brain or a mind even, there are lots of people who now talk about it in terms of, say, the ability to solve problems or make choices between alternative courses of action, or to have some kind of goal-directed behavior, to seek a steady state when the environment's been perturbed. And in these more general terms, you can think of almost all organisms as having intelligence to some degree. And I think it's much more helpful to think about it like that, to think about, say, a single cell has a more minimal intelligence, more minimal cognition, but it's undoubtedly a sensing body. It's undoubtedly managing data streams from its environment, integrating those and making choices between alternative courses of action. Even an E. coli bacterium can go away from something or towards something that it wants to go away or towards from. So in these more expanded definitions, then intelligence is just a general principle of life, and you can have more or less complex amplifiers of intelligence. And then consciousness is a separate question, of course, the degree to which organisms are aware of their experience. But the intelligence thing has really opened up recently, and I think it's a great development.
That's so cool. So we're pretty much out of time, but I just want to say, if you guys haven't read Entangled Life, you definitely should. Merlin is a musician, but so is your brother. And I have to say, am and have always been a big fan of Cosmos, and it was a huge and very strange recognition when I found out that you guys were brothers. I was like, holy cow, two people I'm really excited about and really respect what they're doing happen to be related, because, you know, how many people have your last name? I think your folks have some interesting stuff going on too. So thank you so much for joining me. I could go on. There was things I didn't get to, like bioelectricity, and I mean, just it goes on and on. So maybe some other day we can chat again. But thank you so much. And, you know, if you see your brother, tell him I say hi. Big fan.
Sure.
But yeah, would you be willing to like hop on for maybe like 20 minutes of Q&A? Because I think we've got a lot of interesting questions, and yeah, we'll get to it. So let me post this, because fingers crossed, Instagram has screwed me and deleted a lot of these interviews I've done, but this is a really good one, and I appreciate you so much. So let me try to upload this, and I'll start another one and let you know. Okay.
Okay, great.
Thanks very much.
Thanks for having me.